All right, good afternoon. Let's finish up lecture 21. We are at lecture 21. It seemed like a number that was too big. We are getting to the end of biochemistry, and so you're making it. And let's get a little closer to the end by finishing up chapter 11. So remember that we left off with the idea that different membranes have different compositions. And you can look up and down, and it's interesting. You can see a plasma membrane, for example, has a lot more cholesterol. And it also has a good number of sphingolipids because it's on the outside. There are different membranes, like lysosomal has sphingolipids. There are sphingolipids like everywhere. They're not fully associated with the outside. But they seem to be associated with both of these. Mitochondrial membranes are pretty different. Notice that mitochondria have this red cardiolipin in them, especially the inner mitochondrial membrane. We'll get back to that at the very end of this lecture. You can come up with observations. You start with observations about what's unusual and what's in common, and then you start to try to explain those observations. And then you can look at the inner monolayer and the outer monolayer differently. And the thing is, these are defined concentrations. These do not change that much over time because it's very hard for a lipid to go from one to the other. It doesn't happen. It has a high transition state, not impossible, but very hard. And the high transition state means that it just doesn't happen very much. And so you have a defined difference between the outer and the inner monolayers. And you can see right here, at the bottom you have all the phosphatidylinositols, and those point in, not out. That's because they're actually used in cell signaling. We're actually going to see those in chapter 12. Phosphatidylcholine and sphingomyelin, on the other hand, they point out. And I believe that are those necessarily glycolipids? I know that if we have glycolipids on this list, the polysaccharide glycolipids would be mostly pointing outward. Inositol is different. It's a different sugar, and it's pointing inwards. So it's doing something differently. It also works as a monosaccharide, not as a polysaccharide. So just realize glycosylation in general happens more outside. Something different is going on with inositol, and we will talk about that. Those actually send signals to the inside of the cell, whereas those with phosphatidylcholine will tend to send signals to the outside of the cell. I think I said that right. I had it right printed. So because there's such a high transition state, flipping at the top, it shows a diffusion from one leaflet to the outer to the inner. And it will happen, but it's a half-life of days. It happens very slowly. Now, of course, lateral diffusion, staying in the same leaflet but just moving around, is very fast. And the lipids can move like up to a micrometer per second. What's interesting about notice I said the transition state of flip-flopping from one leaflet to the other was very high, and therefore it was a very slow reaction. I said that deliberately because there are enzymes that actually will catalyze this reaction, that actually will stabilize the transition state, and they are called flip-flopases. And in fact, they actually, if they work in a direction, there are some that only flip from outside to inside, and some that only flip from inside to outside. Both of those are powered by ATP, and they have to be because you're moving things in a particular direction. And one's called a flipase and the other one's called a flopase. But then there's another kind called a scramblase. It doesn't cost any ATP at all. What it does is it randomly just moves lipids from the inside to the outside at the same time it moves one from the outside to the inside. Because this is a random movement, you're moving two compensating lipids in different directions, it actually doesn't require ATP. The drawback is it just randomly scrambles all of the lipids inside and outside. And so if you want to do that, it's like the chaos agent when it comes to the enzymes that catalyze this. Trans-bilayer diffusion is what it's called, although I think it's more interesting to just say it's a flip-flop. And so there's membrane scientists, and it's an interesting and growing field. And there's one story from a membrane scientist that I want to show you that I got a couple years ago. So this is you can actually measure the absolute composition. This is actually a mass spec technique. They're looking at mammalian plasma membranes and the two leaflets, inner and outer. And they added it up, and it didn't add up for them. They realized that the inner leaflet had 50% more lipids than the outer leaflet. So they have a long thread about how they looked at this. But what's really the take-home of it is that this is a real thing. The inner leaflet actually does have a lot more lipids than the outer leaflet. Hopefully I said that right. Again, it's printed right. Sometimes I say things wrong, right? But the inner leaflet has more lipids than the outer. Because we assume, like, all the pictures in your book show that they have about the same. No, it's actually, for mammals and for the plasma membrane, there's way more in the inner because of cholesterol. Okay, and this has major consequences. This is what it looks like, because if there's so many fewer lipids, there should be a huge gap between them. After all, we have to form a bilayer, right? But cholesterol actually can flip back and forth between leaflets, and if you look at it, it doesn't have a polar head group in the same way that regular phospholipids do. So cholesterol can flip back and forth, and it can actually fill in the gaps and help with the whole dynamic nature of the membranes. This is really cool. I love finding out when the textbook assumption is wrong. So when there's fewer phospholipids, cholesterol sort of hops over to fill in the gaps. And they're showing the cholesterol in green on this particular issue. If you have like 40% cholesterol, it allows the imbalance to take place. If you have only 10% cholesterol, then you start to get bulging lipid membranes. And you have a picture of that on the top left here. If you take out the cholesterol, the membranes start to not work. And they've actually tested this. They have the simulations that they've done. Then they tested it by taking out the cholesterol, and they observed exactly what they expected to see. So this is another reason why cholesterol helps with the flexibility. And in fact, this might explain the observation that when cholesterol was invented/evolved, that suddenly life got a lot more able to do more things with its membranes. So one interesting thing that I want you to think about, cholesterol prefers saturated lipids, which are highly enriched in the outer leaflet. This helps it to flip across to the outer leaflet to help make up for the imbalance. So cholesterol is enriched in it, and they actually showed that this happens in real life. They came up with a new assay. Very cool paper. A lot to take apart here, but the main thing is the textbook is wrong. But it's right if you include the fact that cholesterol can move around really fast. And there is a scrambling that happens when you have this going on. I just think that's a cool story, but I want to ask you, the thing that they say, it strongly prefers saturated lipids. Why would cholesterol prefer saturated lipids? What about its structure makes it do this, which actually ends up with it working the way it should? I'm not going to answer that question. I might ask it on a test. We'll see. So another thing that you learn in different kinds of science is that you have two basic kinds of bacteria. You have the gram-negative and the gram-positive ones. You can see the gram-positive includes pneumococci, it includes streptococcus, it includes other things, listeria, other things that you've heard of, clostridia, and bacilli. Escherichia coli is actually gram-negative, and so you see on top, has to do with how they stain a dye, and this means it has to do with what's on the outside of the cell, which means it has to do with lipid membranes in some way. So the lipids are actually attached to sugars in gram-negative bacteria, and there are peptides attached to the sugars in gram-positive bacteria. In fact, the gram stain, we mentioned it before, gram stain actually stains peptides. And this means that gram-negative bacteria are going to have a different bacterial membrane structure. Here's a picture of what it looks like. You have the phospholipids at the bottom, like usual. Then you have something called lipid A and a core saccharide, and then they have the O antigen, which is more saccharide. You see how much sugar they have on the outside. They're basically very sweet. And so you have a special lipid, and then two special saccharides that are attached to that special lipid. And that's what gram-negative bacteria have on their outside. Gram-positive bacteria have that cell wall, the peptidoglycan that we mentioned, and this is what they look like. It looks rather different. You have the cell wall that has this rigid scaffolding. This is shown with the cell wall on the bottom, by the way. This is a very nice picture, though, because it's shown in green. You can see they have the peptides sort of as a scaffolding holding it together. And these are pretty major differences between sugars and peptides, and they're going to act very differently, and it's a good classification system that we've come up with. So one of the main things about membranes is that they need to be fluid at the temperature at which they function. And so this is why, to explain this, we have a fluid mosaic model that explains how membranes move. This is one of the major concepts that you need to finish up Chapter 11. So the mosaic is the other bits that are not lipid. Like the fluid part is the lipids, and then the mosaic part is the bits of protein or maybe sphingolipid rafts swirling around. Remember how we said things are swirling around? You have islands of protein or of rafts of other lipids that are moving around in there. The way this was shown was with a pretty cool experiment. And if I think it's pretty cool, I'm going to explain it to you and want to think about you explaining it back to me. You take a regular cell and you put a fluorescent probe on its outside. There's a lot of probes that will do this. A lot of fluorescent probes will actually be bleached when you zap them with a laser. They'll basically be not fluorescent anymore. And so what they did is they put this fluorescent probe on the whole cell. They zeroed in on one part of the membrane, and this is normal temperature cell, so everything else was normal. Then they zap it with a laser, and they create this bleached spot. But then as they sat there, and as you sit there, you can time how long it takes. You will see fluorescent unbleached lipids diffuse into the bleached spot over time. And you can measure the rate that the fluorescence returns. The fact is, this shows you that they're able to diffuse. The lipids that are unbleached are able to diffuse into the bleached spot from outside. This shows that they're fluid. They're basically liquid, but they're liquid in two dimensions. So, and this makes sense because membranes have to flow. You have to have the lipids be able to flow past each other for life to be able to respond to its environment. Otherwise, it's just like a solid between life and its environment, and it's not able to respond. And so you can have a sort of, if a membrane gets too cold, then they'll basically freeze up. It'll literally become a solid lipid state, like a solid lipid rather than a liquid lipid, and then life will not work anymore. But at a certain temperature it will melt, and you'll have the liquid disordered state shown on the bottom. There's a very distinct transition between this, and we've seen this with proteins, we've seen this with DNA, and now we see it with lipids. There's a temperature at which things melt, bonds break, and you move from one phase to another. And so membranes will flow differently depending on what lipids they have in them. This is a cool figure. This is, of course, the details are just too much to remember. But I want you to look at this from left to right. Start on the left and look at that fluidity part of it right there. You have unsaturated and saturated lipids, and the saturated ones are going to be more solid. The unsaturated ones are going to be more liquid. And so at whatever the temperature is, you want the right mix of saturated and unsaturated so that it is still holding together well, but it's liquid enough for it to diffuse. In other words, if you need to work at a colder temperature, the microbe or the cell membrane will survive better if it has more unsaturated lipids to make it more liquid at that colder temperature. So what matters is whether you have unsaturated or saturated. It also matters whether you have thin lipids or thick lipids and how many sterols you have. Sterols are kind of thick. And it matters how things are packed together. It matters surface charges. The bottom one is actually the one I want to make sure you know, because you can have different charges on the head groups. We showed that on the previous table. So if you have negatively charged head groups next to neutral head groups, then they will actually, like the negative charges, will repel. When it all comes together, you get the figures on the right, where you have the ER membrane, and then you have the plasma membrane on the bottom. These are actually different. The ER membrane is a little bit thinner. It has lipids with shorter tails, whereas the plasma membrane has longer lipids with longer tails. As a result, a transmembrane protein can cross the membrane with just 20 amino acids in a helix that has hydrophobic stuff on the outside. 20 amino acids is sufficient to cross the ER, but 25 amino acids is needed to cross the plasma membrane. There's also differences in charges, differences in cholesterol, and it's just a fascinating area that you can look into. And it's important to how life works, because saturation matters. Not just all natural saturation is going to be cis bonds, which will be kicking out. But if you have trans fats, it's actually going to solidify your membranes a little bit, right? Because they are going to be straight up and down rather than kicking out like the cis natural ones. The trans fats are going to be artificial, and they are going to keep the membranes more solid than they would be otherwise if they had natural cis bonds, double bonds in there. And so this was actually used to explain some experimental results that were along the lines of olive oil, giving olive oil to rats and asking what their blood pressure does. So they fed the rats cisoleic acid, or transoleic acid in the olive oil, or soybean oil which had no oleic acid. And they found out that specifically the cisoleic acid rats, they had lower blood pressure. But the transoleic acid rats had higher blood pressure, just from that one difference of eating more trans fats. So they looked into why, and they did find out that what you expect is true. The cis bonds will make the membrane more liquid. And so blood pressure will actually go up, and it has to do with signaling. It's not a straightforward pressure thing, but the cells are more receptive to signals if they are more liquid. This shows you that the membrane needs to be liquid, and you need a certain amount of unsaturation to be able to make it more liquid. This is shown when you grow E. coli at different temperatures. You'll see that it will have more fatty acids. At colder temperatures, it's going to have more unsaturated fatty acids. At warmer temperatures, it's going to have more saturated because it needs to maintain its fluidity. And this is the data that shows it has all the different kinds, but you can reduce it to one number. The bottom row says ratio of unsaturated to saturated. You see that's a very clear ratio, very clearly different at 10 degrees than it is for 40 degrees. And we can understand why. Because membranes are fluid, they do all sorts of things, and these are all the things you talk about in cell biology that will tell you, if you wonder how does a cell do that? Well, it's because of biochemistry at the bottom of it, because non-covalent interactions are arranged in 2 dimensions by the chemistry of the lipids. Very cool. And so this includes how viruses infect, it includes how endosomes fuse, it includes how sperm and egg fuse, how plants form small vacuoles, and how 2 cells divide. All of these have to do with the membrane being a fluid the way it is. These all come down to when you join 2 membranes together, they dissolve into each other, right? That's what happens when you have a fusion event. Also, if you poke a hole in the membrane, it will seal it up because of these properties. Membranes are very cool materials if you really look at them. So you can also do studies where, remember how we did studies where you put fluorescent probes all over the outside of the cell and looked at a microscope? Well, you can do that on one protein. You can fluorescently label a protein, you can see how it moves around, and as you can see, you can sort of start over on the right side here. And you will start with, you see the protein sort of follows a certain random path. It doesn't look like it's doing much of anything until you look at it by time. The protein will diffuse all around in the purple area, and then it will jump to the blue area and it will diffuse all around in that, and then it will jump to the green area, and finally it will jump to the yellow area on the far left, which on your direction is over here, right? This is interesting because it's not purely random. It's random within a bound. So like the protein seems to be fenced in somehow. And so I looked into how to do this. It turns out the cell does have structure underneath the lipid membrane that creates these areas where the protein will tend to stay in that area, but then it can jump from area to area, as the case may be. It's not super high energy. It's just kind of high energy for it to jump from area to area. There are some proteins that never move. These proteins don't diffuse at all because they are tied down to the cytoskeleton. And so the cytoskeleton is what these proteins don't move on. If they are not tied down in the cytoskeleton, actually the cytoskeleton also defines the fences that the other proteins can hop over. So the cytoskeleton means that there's this underlying skeleton of proteins. And realize that it's a cell skeleton. It's made of proteins, fibrous proteins that form this sort of network. It looks like a soccer ball a little bit. That you have these different areas underneath. If a protein's tied down, it doesn't diffuse. And you see that you can have some, like the chloride bicarbonate exchange proteins are tied down. I think that actually has an effect on some diseases. There's an association with the cell membrane. And also the cytoskeleton, I should say. The cytoskeleton can be moved around deliberately by the cell to move these proteins that are attached to it one way or the other. You can actually, proteins that are attached to actin are what makes the fence that keeps these other non-attached proteins sort of in their own area. It's a little fence, but they can squeeze through from time to time. And that accounts for the behavior that we saw on the previous slide. It's very cool. And I was just reminded of that when I saw this image. This is GPS tracking of 6 different wolves in Voyageur National Park. I think that's Canadian. Okay? If you know where it is, let me know. But there's a national park. They had some wolves. They put GPS collars on them. There were 6 wolves, and look at how those wolves each stay in their own area. In the same way, lipids will diffuse within their own area, kind of like a wolf will diffuse in its own area. And then it will hop, the lipid can hop from area to area. If a wolf tries to hop from area to area, it's going to run into another wolf. And so they have this self-organizing domain kind of behavior. I just think this is one of the best ways in which my lecture actually overlaps with what Dr. Long talks about. So I had to take this chance to talk about it. Proteins diffusing in a monolayer are kind of like wolves establishing their territory. But that's another topic for another day for me to talk to him about. So what can you do when you try to take apart membranes and look at them? Well, one thing you can remember the rule that sugars are already on the outside. If you have a protein that goes all the way through the membrane, it has a transmembrane helix or other transmembrane kind of sections, then you have an outside and an inside, and then you have a membrane part. And that protein is not going to flip over, so these are going to be very well defined biochemically. So, for example, you can do things like you can apply a protease to a cell protein and then purify that protein and run the gel, and you can see where the protease cleaved the protein. The protease can only reach the outside, and that can tell you which part of the protein is outside as opposed to inside. You can also look at the sequence, look for glycosylation sites. Remember, those only happen at serines, threonines, and asparagines. And if you have those glycosylation sites and the signals that make them only in one domain, you can tell that that is the outside domain. Here you have the amino terminus is outside. The amino terminal domain is on the outside of the cell. The carboxy terminal domain is on the inside. You can also tell where it's in the membrane because that's where you have a ton of hydrophobic residues that will interact well with the membrane. And if it's an alpha helix, it'll be a certain distance because we know how many turns per helix, we know how many amino acids, and we can actually look for these patterns in protein sequences. So we don't actually have to make the protein to have the guess that it is a membrane protein. Yeah, notice how all of those amino acids are hydrophobic. Be a good test question to ask you based on the amino acid names and structures, which you should still know. you should be able to even look at the sequence yourself and say, hey, there's a hydrophobic region right there. So just remember that some membrane proteins are going to go all the way through. They're going to have a hydrophobic region. Some membrane-associated proteins are more weakly associated, and they only sort of sit on the membrane. They only visit for a while. And there's a big difference between these two kinds of proteins. The ones that just sit on the protein are called peripheral, and they're only going to interact with the charged head groups, maybe phosphates, maybe the other groups that they have. But they are not going to have big stretches of hydrophobic residues, and they're going to be pretty easy to remove it from the membrane and purify them out. If you can only remove a membrane, a protein by using detergent, then you can tell it's an integral protein. Peripheral proteins can be removed by just changing the pH or by adding an enzyme that will kind of cleave them. And notice right here, there's a phospholipase C shown here. There are some proteins that are peripheral proteins. They don't have big hydrophobic sections, and yet they stay embedded in the protein until you add phospholipase C. This because they actually are attached to a little lipid anchor, which is very cool. There's a particular phospholipase that will destroy their anchor, and then they'll be set free. The anchor shown here is a GPI anchor. And so we want to mention what that is. And these are the kind of things that can do protein chemistry experiments in a membrane context. So here's different kinds of integral proteins. You have an integral protein. These all have big alpha helices that are hydrophobic. Even the monotopic one that they show, and that one's kind of weird. It looks like it might be peripheral, but no, it stays stuck in the membrane because it's got a whole hydrophobic helix that is holding it in. There's pictures of what these all look like, and I just want to say COX-2, which is involved with the aspirin and the steroidal, the non-steroidal anti-inflammatories, that is actually the enzyme that is targeted by those. That is a monotopic integral protein. But the point is, it's very hard to remove. You can only remove it with detergent. Peripheral proteins can be removed by pH, by urea, by carbonate, by little things that change, just little things about if you unfold it a little, it'll fall right off. And they'll also be removed by chelating agents. Chelating agents will bind positively charged ions and take them away. So this is an interesting thing. They found out that calcium actually helps some peripheral proteins bind. You chelate the calcium, take it away with a chelating agent, and then the protein falls off. Very cool. And so you can talk about the different amounts of embedded nature for the monotopic proteins that have sort of the sideways helices. Most of your textbook will be talking about the polytopic or bitopic proteins that have membrane spanning helices. So here's an example of a protein that actually is peripheral, and it's attached to the protein through calcium groups. So it actually has, I thought I showed you the charge there, calcium's plus charged. It looks like it has phosphatidylcholine there because it looks like an N with 3 methyls on it, which means it'd be positively charged N's, positively charged calciums. You're going to have a lot of negatively charged amino acids in the annexin protein that you expect to be able to have charge-charge interactions that hold it to the membrane. If you take out some of the calciums, you take out some of the charges that are holding it in. Very cool. So the lipid anchors are actually kind of cool themselves. There are lots of different kinds. And again, it's the general concept that you want to know. These lipids are covalently attached to the protein. And you can see that there's covalent bonds. There's like a thioester or an amide bond or even a thiol bond down on the different palmitoyl, myristoyl, and farnesyl groups. Again, you'll probably see these in a cell biology context. I'm not going to expect too much specificity for biochemistry, except for you to know these are very tightly bound lipids. They are covalently bound, and it takes a specific enzyme to cut that covalent bond. So that means that they're held into the membrane pretty well. They're technically peripheral because they are only held in by that lipid anchor, but it does a pretty good job of keeping them in the protein, keeping them in the membrane. The other thing is you have different kinds of anchors for different sides of the membrane. The ones I just mentioned, the three I just mentioned, are all inside anchors. They're single fatty acids added on. But there's a special kind that's outside called a GPI anchor, and I just want you to take a look at this. It's got two lipids, and then it's got basically a phospholipid with two tails. Then it's got inositol, and it's got a bunch of sugars, and then it's got a covalent bond to a phosphate. So that is bound to the protein itself. It's a GPI anchor, and actually the proteins I worked with in grad school were immune signaling proteins on the outside of cells, and some of them are GPI anchored. That's how they're held into the cell. So I have a special place in my heart for the GPI anchor. It's important to my proteins, so it's important to me. So just remember there are different types of integral membrane proteins, and you can have all these different types of the kind that span the whole membrane. And I do want you to know these because it's not too much to know. I want you especially to know types 1, 2, 3, and 5 because these are pretty straightforward. Types 1 and 2 are just one helix each. Type 1 has the amino terminus on the outside, and so you can think of that as being sort of the simplest case for us because we always start with the amino terminus. If the amino is outside, it's type 1. If the carboxyl is outside, it's type 2. Then you have type 3 where you have a bunch of helices that span the membrane, but there's multiple ones of those. Then you have type 5 where they're not all in one chain like they are in type 3. Type 5 has multiple chains, and it's held together by the membrane itself, sort of holding the chains together. And this is why it's important. Bacteriorhodopsin, again, we're going to cover this in the last chapter. Bacteriorhodopsin is a membrane protein. In fact, I can just show you the picture, and I can say, which type is this? Well, it's a type 3. It's all one chain, multiple helices. But actually, I can ask you, I can not show you just the picture. And by the way, when we get to chapter 12, we're going to talk about how this opens a door for protons to cross the membrane, and it's how bacteriorhodopsin works. So it's important for us to have this in our minds, and we will follow up on this soon. But if I showed you the structure, you should be able to say, oh, that's a type 3 integral protein. But I can actually just show you a sequence, and you can say, oh, that must be a type 3 integral protein, because the sequence has 7 hydrophobic stretches that are all about the same stretch, all about exactly the number of amino acids that you need to cross a membrane. And so you can do what's called a hydropathy plot, where you have if you go residue by residue from left to right, and you say whether it's hydrophobic or hydrophilic. You can sort of add up adjacent residues that you can have context effects as well. But basically, this is saying, I see a bunch of valine in the part labeled part 1, purple 1. We maybe see valines and, you see phenylalanines and valines and isoleucines and stuff like that. And then you get to residue 50 and you see a lot of hydrophilic residues. But you see these long stretches of 20 to 25 hydrophobic residues, and you see 7 of those, and that turns out to be a signal that it's a bacteriorhodopsin-like protein. In other words, you don't need a structure. You don't need a tertiary structure. All you need is a sequence or a primary structure. So if you look at this on a hydropathy plot, you can say, okay, what do you conclude about this? Which type of integral protein is it? You can pause and go back if you want to review the types. You can say right away it's either, you can rule out type 3 because you only have one possible helix going across. I guess it could be type 5 if you had multiple ones of them that came together. And it could be type 1 or type 2. You need more information, but this is evidence for just one membrane crossing helix. Glycophorin is that protein. So we can look at these and we can say membrane crossing helices, they're going to be all hydrophobic. Outside ones will be hydrophilic. What about ones that are about at the level between outside and inside? And so look at this. You have charged residues colored blue, tryptophans and tyrosines colored red and orange, and then all other residues are colored gray. And so if you look at this, on the outside you see a lot of blue. Charged residues are outside, and technically they are inside the cell. We're talking about inside the membrane here. So charged residues are outside the membrane. At the water-lipid interface, you actually have a lot of red and orange. So that means that you have tyrosine and tryptophan preferentially. And that's interesting because both of those look like they'd be happy in either water or lipid. And you put them at the place where they're going to be contacting water and lipid. It's very cool. You can make sense of these patterns if you just know what the amino acids look like. And so what's inside the membrane? Uncharged residues. And also you can see the rule is not 100%. You can see a few patches of blue in the membrane, but remember biochemistry is not 100%. It's usually 90-95%. In fact, that's why A is 90%. You can still miss 10% of the class and get an A. It's that kind of thing, plus extra credit. So just remember about how you want to remember how things move in the membrane. And there's some interesting aspects. It looks like proteins have adapted to move in the membrane in different ways. You can have a protein embedded in the membrane that will conventionally diffuse. We talked about that before. But then there's some proteins that seem to diffuse faster through the membrane. They're trying to figure out why. These proteins actually have they interact with the membrane. They sort of wrinkle it up. They have membrane spanning helices that don't necessarily go all the way through, and they sort of bend the membrane to make it more fluid around them. That means that they actually can move faster. It's a little bit like an icebreaker, the way the icebreakers work, that they have like a front edge that will actually, and I'm talking about the ship that's an icebreaker, that will actually have the front edge that sort of disrupts the ice so they can move faster. This is kind of like that. It's kind of like an icebreaker for lipids. And they can diffuse faster. I just think this kind of membrane research is really fascinating. It's a protrusive shape that distorts the shape of the membrane, just like an icebreaker ship has a protrusive end that breaks up the ice. And so just remember, it's not always about proteins. You can have thick regions of sphingolipids. They sort of dissolves like, and they sort of fit well together, so they form these what are called microdomains or lipid rafts. And you can see actually they are thicker. They are little islands that are sort of jutting out of the cell membrane. This is a microscopy picture of the surface of a membrane right here. If you look at it from the side, you can see that they will have different concentrations of cholesterol, and also those lipid anchors like to interact with sphingolipids. And so you'll tend to have GPI anchored proteins or other anchored proteins on the inside or outside as the case may be. What's really cool about this is they found out what happens with these. Aren't they sticking out too much if they're too thick? Well, they actually do stick out too much, but that means that they tilt. And they tilt all in one direction. They all tilt over, and this means that they actually will spin in certain ways. And so these lipid rafts will create these vortices, like whirlpools, on the surface of the cell, and that might be where some of the ocean current type things that we got came about. It looks like the cell has this complex pattern of moving parts, and it even has like these lipid rafts will be spinning there. And of course, as you lower the temperature, they're going to spin less, they're going to freeze. And they saw exactly that with this study. If you have a hydrophobic mismatch, which means that you have things that are too tall for the membrane, they will create cool dynamic effects. It's not an accident. It looks like it has a reason for being that way. And the final, we can also talk about things like what happens when you electroporate a cell. If you've ever electroporated to get a plasmid in a cell, we do that sometimes in lab. You literally are applying a charge and you're making pores. You're making big open spaces. And so you can have computer simulations of the lipid membrane that tries to say, what does this look like? And I don't know, this makes sense to me. You know, you have the lipids that, you have the holes that are forming, and maybe they form on the edge of the raft. So I think that might be what they're showing here. Yeah, you can look over, and as you electroporate it, you create all these little holes for a second. But then the lipids flow back in, and the membrane heals. Life goes on. You can make doors in the membrane. You can make short little pores in the membrane by applying an electric charge. But if you're a cell, you can't do that. But you can make a protein that will make a big hole in the membrane. And there's even a beta barrel that will basically make a hole in the membrane like this. And that will actually make a pore big enough to let an entire small protein through intact. And you have things like the nuclear pore complex. These can get really complex. Really cool cell biology stuff. Here's some examples of beta barrel type proteins. And you can see that we have all the way on the right, look at that one. That's a toxin, Hemolysin toxin. What do you take that word apart? What do you think it does? Hemolysin. It lyses hemo, blood. So I think this lyses red blood cells. In fact, I'm so sure I've never looked it up. I think I'm just reading well. So we'll see if that, I might be proved wrong, but I believe this is a toxin that disrupts red blood cell membranes. What it does is it punches a hole in the membrane. You can see the top view. It's a pretty big hole when you look at it from the top. From the side, you can see it's a beta barrel that is in there. Yeah, and how hemolysin works, it actually works as several subunits that are soluble. They're in the format of A, and they have this beta barrel part, the beta hairpin, is folded up. They dissolve around to get to the cell, and then the orange hairpin reaches out and it joins into the cell, and it joins with all these other ones. It makes 7 of these hairpins come together to make the beta barrel, and then that makes the pore. I think this is really an amazing protein, but it sounds like it's bad news when it comes to disease. Maybe we can get in its way. We can come up with a therapy that prevents hemolysin from breaking up our cells. The FepA protein was another one that was shown. It's big enough, it has a whole protein, and it has a column of water. Remember that we always have water. We show it like it's an empty space. But in the cell, it's always full of stuff. So they show you can simulate the water going through the membrane, going through the protein. Very cool computer simulations here. And the thing you've got to remember about proteins is they're very solid. They're big chunks. They're the mosaic in the fluid mosaic model. And so the lipids right next to them are going to be sort of solid as well. They're going to be sort of stuck to them. And in fact, if you get certain kinds of crystal structures, once you apply a detergent, and you can apply a lipid-like detergent, and you can see the lipids, there are still some lipids stuck onto the protein after you remove it from the membrane with the detergent. So here's sheep aquaporin, and these are called annular lipids. It's kind of a cool structural effect, so you should know that word. You see, sheep aquaporin has these annular lipids, and it's kind of like an imprint of the membrane. You can see where the membrane was, because you see where the membrane is still stuck to the protein. For the ATPase, which is going to be a topic in Biochem 2, you can see that there's annular lipids on the inside of the pore, and this shows you where the membrane fits into the ATPase. This is the V-type ATPase. We talked about how it's a big, complex protein. This is part of the part of figuring out how does it fit into the membrane. Look for the annular lipids. And this is the last thing that we end up with. I promised you I would tell you what Cardiolipin did. Here it is. And remember, it's a two-headed lipid. And what it is it's found in mitochondria. In fact, it actually is an annular lipid for ATP synthase. And it actually is part of what makes ATP synthase work. When you get to Biochem 2, you're going to talk about ATP synthase. It's an amazing machine, but part of it stays in place while part of it rotates. And I think Cardiolipin here is a staple that will actually have annular lipid contacts to different parts of the protein, and it will hold them in place. And it's how the protein does that amazing rotation that it does. When you get to that, try to remember that there's a lipid there that is holding it together. For right now, just see how it is weird. See how it is a two-headed lipid. You have one glycerol in the middle attached to two phosphates. Each of those is its own phospholipid. But this is a pretty tight covalently bound assembly here. And then this can sort of staple ATP synthase together. And that brings us to the end of Chapter 11. Now we move on to biosignaling. We're really going to tie it all together when we get to Chapter 12. That will be Monday before Thanksgiving and Monday after Thanksgiving. So I will see you then.